Just to say that genes do not define everything. Genes are, of course, very important. They make us human. They define many of our traits. 20% of longevity is defined by genes, and everything else is environment.
Welcome to the Fat Emperor podcast. I'm your host, Ivor Cummins. A fascinating discussion today. We're going to be talking about glycans, glycosylation, maybe the glycocalyx as well, some of you may be familiar with, and all about genetic traits that drive disease and also environmental ones and the difference between the two. So we're going to be with Professor Gordan Lauc, and he's a professor of microbiology and molecular biology at the University of Zagreb in Croatia. So great to meet you, Gordan.
Hi, Ivor. Thanks for the invitation and the introduction.
Not at all. And it's I forgot to mention my friend Creon Levitt, who's also kind of a genius, connected me to you. So appreciate there, Creon, getting me connected, because this is fascinating stuff.
Yeah, well, it's quite a novel. Not so many people think about it. So I think it's good to talk about it and let more people into this field, because there is a huge potential.
Absolutely. And I've watched some of your material, so I've an appreciation of it. And as you go through the material, I might pop in the odd time, maybe clarify something, but otherwise it'll kind of be like a talk with an annoying member of the audience clarifying occasionally.
Yep, that's fine.
Very good. Okie doke then, I'll let you kick off with probably the best thing. Get right into it.
Okay, so I'll start with introduction about the glycans and how we can use them as a tool, hopefully, to prevent age-related diseases. So not so many people talk about glycans. They don't even know much about glycans, but glycans are actually the ultimate layer of molecular complexity. And unfortunately, at the same time, the most neglected molecules of cellular communication. And I think one of the best examples is the current COVID pandemics, where the spike glycoprotein looks like this. So all these hairy things are glycans on top of the protein part. And until a couple of months ago, even the director of NIH was tweeting about the same protein without a single glycan. And majority of proteins are glycosylated. And to see the complete picture, to understand how they work, we also have to look at glycans. Unfortunately, majority of people have a myopic view, and they focus on genomics. They focus on some parts of epigenetics, usually methylation, because it's easy to analyze. So it's easy to sequence genes. It's easy to analyze DNA methylation. And things get more complicated when you want to look into proteins, glycans, lipids. But the information content here is much larger. Actually, genes are only a fraction of information about us, and much more is stored here. And actually, the glycoproteome is several orders of magnitude more complex than the proteome. And the simplest way to explain how important it is just to say that genes do not define everything. Genes are, of course, very important. They make us human. They define many of our traits. But majority of complex diseases are only partly genetic. So something like 30% of complex diseases is defined by genes. 20% of longevity is defined by genes. And everything else is environment. What we do, what we eat, what we think is what defines us the way we are at the moment. And I think these two figures are really very clear. These are two pairs of identical twins. Every single letter in their genomes is identical, but they clearly have very different life trajectories. And this is the reason why genetics has actually not fulfilled the expectation of both science and general public. There is a huge technical success. You can easily sequence the genomes. Now it's a couple €100 and you can get every single letter in your genome. But we still do not know how to interpret it. We cannot link genetic polymorphisms with the majority of things in our lives. Of course, there are some monogenic diseases which are clearly genetic, but many things are not genetic. And This is because there are many layers of information between genetic polymorphisms. So this is information in our DNA, and then there's some kind of final phenotype. And there are layers of interaction between genes and environment. And only this together defines the final phenotype. And many of these parts are not encoded in the genome. And one of these are the glycans. So glycans are structural component of nearly all proteins. So majority of proteins, in addition to polypeptide, which is defined by a gene, will have these glycan additions, which form a part of a structure. And the protein functions as a single entity composed of a polypeptide and of a glycan. An analogy I like to make, and I use usually this spike glycoprotein. This is a spike glycoprotein without its glycans, and this is a spike glycoprotein the way it really looks. And if we are trying to understand the function of a protein by looking only at the polypeptide part, it's very similar to trying to understand functions of a bird, studying this unfortunate bird who lost all its feathers. So we can learn a lot about the bird physiology. We can do many experiments on this bird, but we would never see the bird flying because the bird cannot fly without its feathers. The same thing is for proteins. We can do a lot of research. We can learn many things about proteins by studying only the polypeptide part, but we will never understand the function of a protein without including glycans in the story. And this is not easy. This is not easy because there are at least 2,000 different glycan blocks which are being added to proteins. So these different combinations of monosaccharides which form a glycan block, they're added to different positions on a protein, and this expands the complexity of our molecular structures tremendously. Just if you think about it, nucleic acids have 4 building blocks, proteins have 20 building blocks, amino acids, and glycans bring additional thousands of building blocks, which raises the complexity of the glycoproteome by several orders of magnitude.
Yeah, and just on that, Gordon, yeah, just to clarify the enormity of this. So the nucleic acids in our DNA, we've got 4 essentials, and we know what that complexity can generate. The proteins, then, there's only 20, and they can combine together in incredibly variable ways to create the complexity of life. And now you've got the glycoproteins, which are massively important in the function of all of that, but you've got 2,000. So the combinations and permutations are just beyond enormous.
It's immense. So, and it's immense in a way that it even doesn't function as a one-to-one communication. It's a kind of a network structures which interact. It's additional layer of complexity. So yeah, actually
So even like fuzzy logic, chaos theory, you're just into a whole different realm of complexity. And that really does, for me, explain fully for the first time, exactly as you say, genetics loads the guns, but the environment pulls the triggers. So the environment is really driving the bus, even though we would intuitively feel, well, the genetics are what drive everything. They're really only the start.
Genetics is extremely important. It's confounding everything, but environment has tremendous impact. Yeah, and this is exactly my next slide. So when we talk about proteins, there is a gene for every protein, and the structure is defined. The way we got our genes at the moment of conceptions, it determines the structure of our proteins for the rest of our lives. Then comes glycosylation, for which we do not have a template. There is no single genetic template which defines glycans. It's actually a network of dozens of genes which work together, interact with the environment and with each other to define the final structure of a glycan, which will be put on a spot on a protein to perform the function. And glycans are actually responsible for the large part of inter-individual variation. So what determines that we are not all the same are by large glycans. And actually, We know this for over a century because blood groups are glycans. ABO and blood groups are glycan structures which are added to the different carriers. And this is for what Karl Eichsteiner got a Nobel Prize in nearly 100 years ago. And it's not only me claiming that. There are strong policy documents by the European Science Foundation, by the US National Academies for more than a decade, claiming that glycans are directly involved in the pathophysiology of every major disease, and that additional knowledge from glycoscience will be needed to realize the goals of personalized medicine and to take advantage of the substantial investments in the human genome and the proteome research and its impact of human health. So these are the statements endorsed by the US National Academies. But people still don't analyze the glycan sufficiently, primarily because this is difficult. So we all try to choose the easy solutions. And for science, easy solutions is look at genetics, now look at epigenetics, now more microbiome, metabolome. These are things which are relatively easy to measure. While glycans are very complicated, they are nonlinear structures. There are many chemical steps which we have to do in a lab to measure them. And this is not so easy to do in high throughput. Now, there are only a few labs in the world which really do high throughput glycomics. And my lab is practically leading this field because we analyzed over 150,000 different glycan samples from some of the best genotype and phenotyped human cohorts in the world, including, for example, 20,000 samples from twins in Twins UK database, which was set up by Tim Spector, a number of clinical studies, epidemiological studies, altogether 150,000 people with a lot of other information, which we can now combine with glycans to try to understand the biology behind many diseases. And one of the things we are focusing on is the IgG glycome. Immunoglobulin G is the major weapon in our arsenal against pathogens. And its effector functions, so what happens after the IgG binds to antigen, are by large determined by glycans. So glycans are here in this so-called FC domain, which determines binding to the different receptors, which then triggers different branches of the immune system. And there is a lot of research done by multiple scientists showing that if these glycans are different, they will activate different branches of the immune system, and there will be different consequences. For example, the same immunoglobulin molecule can be pro-inflammatory if it ends with these glycan, but if you add galactose and salicylic acid, the immunoglobulin will become immunosuppressive. It will suppress inflammation. So the change in glycosylation can completely change the function of immunoglobulin.
So that switch between inflammatory and anti-inflammatory is mediated by these glycan structures that fundamentally switch the functionality of IgG. So that's pretty profound.
Immune system is tremendously complex. So there's more than immunoglobulins and more than immunoglobulin glycans. But yes, glycans and immunoglobulins are one of the mechanisms which will convert IgG from pro-inflammatory to anti-inflammatory molecule, which I will come back in 2 slides. One slide before, I will just say that what we are doing is that we are using our high throughput technology to look whether people are different in the way we glycosylate immunoglobulins. And one of the clearest images we created is this is just 6 random individuals, different color is a different glycan. If you just look at it, people are different. So we are not all the same. We are not just one Homo sapiens and the other Homo sapiens. We are different in the way we glycosylate immunoglobulins. And this is functionally important. So for example, this molecule here is the main pro-inflammatory glycan. Some people, median level is around 20%, but some people will have 5% of it. The other people will have 50% of this structure. While these here are more those glycans which suppress inflammation. And there is a known therapy called IVIG, where you inject immunoglobulins from generally young and healthy people. And this is suppressing inflammation, and it's being used in a number of different inflammatory diseases. There are even some people who take it just to feel better. And this IVIG, by large, works through IgG glycosylation. And now pharma companies are trying to make IVIG with better glycosylation to be more effective. But now when you think about normal people, and this is the pro-inflammatory one, if somebody has 50% of it, it's very pro-inflammatory. But if somebody is having only 5% of this glycan, it's like this person is continuously receiving this IVIG therapy. It's suppressing inflammation. And this is something which is, we don't know how is this regulated, but one of the dreams I have that will identify a small molecule which will convert IgG glycosylation, make it more immunosuppressive, though in cases of inflammatory diseases, acute inflammation, you can give this small molecule, convert the IgG, and suppress inflammation in this way. Because IVIG is not an easy treatment because you have to take blood from many people, isolate IgG, and inject this IgG into patients, and the amounts are huge. So it can go up to couple of hundred grams per patient. So you have to get infusion for hours. It's not an easy therapy, but it works.
Wow. And this, where you are on this scale, pro-inflammatory or tending towards anti-inflammatory, as you describe, so genetics have a part to play for sure, but the environment is manipulating this and dictating this also to a very large extent, right?
Yes, and this is what I'm going to talk about in the next couple of minutes. So the first thing, to be able to look into that, you have to have tools. So what I wanted to say here, people are different, not only in the way they look, but also in the way they glycosylate IgG. But to be able to see these differences, you need tools. And in the last decade or so, a number of different technologies have developed which can analyze glycans in high throughput manner reliably. So we can accurately measure glycans in a large number of people. And we are doing it routinely. So we did it so far close to 80,000 people, looked at IgG glycome. And one of the first things we learned is that heritability is approximately 50%. So heritability is a measure how similar we are to our parents. And what you can see on these images by Craig Gibson, this is the father and the son. If you overlay the faces, you'll see the same face. So facial expression is very heritable. IgG glycome is approximately 50% heritable, meaning if I'm here in this low inflammation area, my parents would probably be somewhere around here too. But only about 50% is genetic. Everything else is something else. And one of the things which is very important is age. So by looking at those large cohorts, we learned that these anti-inflammatory glycans convert to pro-inflammatory when we age. For example, this is one of the major pro-inflammatory. As we are getting older, it is getting higher. So these are 4 cohorts, each over 1,000 people. Blue are men, red are women. And you see that the pattern is more or less similar. And these bumps you see in females, it's around menopause. So these are the pro-inflammatory. They go up. And these are the anti-inflammatory. They go down.
And that's quite striking because many things change with age, many biomarkers, metabolite levels, etc. But this is quite dramatic compared to most of them. This is a huge change.
This is a huge change. And the second thing is, this is also driver of aging. So this is not only a biomarker of aging. This is actually a molecule which, according to this inflammaging theory, is causing molecular damage because as we are getting more pro-inflammatory, we have more and more of those small, low-grade chronic inflammation events. And each inflammation is, neutrophils come in, they kill everything, you have to rebuild it. And this rebuilding is always causing small mistakes, and then you have scars, molecular scars, and actually our organism is aging. So when we learned this, we said, okay, can we predict age from glycans? So we put it in a model, and we could guess a person's age plus minus 9 years. And this was in sometime in 2013 when we first published this. And at that time, this was the most accurate measure of chronological age. So we were thinking to use it in forensics. So we even published a paper in International Journal of Legal Medicine, where we were thinking to, you have a dried blood stain on a crime scene, you can see how old was the person who was bleeding. But then in the meantime, epigenetic clock was developed, which is way more accurate for chronological age, because Steve's epigenetic clock, Steve Horvath's epigenetic clock, can guess a chronological age plus minus 1 year. So now in forensic purposes, it's mostly epigenetics which is being used. But what is interesting for our GlycanAge is not guessing the chronological age, because there is no real value in that. The real value is, okay, you have a dried blood stain, how old was the person, or you have a legal immigrant, is it minor or not minor? These are maybe the two important things you can do with guessing chronological age. Majority of people know their chronological age. So if I do a test to tell you how really old you are, what is the value of that? But what we were asking is, if my GlycanAge is, for example, 60, and my real chronological age is 40, what explains the difference? So what explains the difference between my real chronological age and the age estimated by glycans? So we looked into our large population cohorts and found that the things that correlate mostly are high insulin, high fibrinogen, HbA1c, BMI, triglycerides, glucose, all the unhealthy things. So unhealthy lifestyle is making my glycans look older than in people who are living healthier lifestyle. So then we got an idea, wow, maybe our glycan age could be something which will tell us how healthy we actually are.
And just looking at that list there, Gordon, I mean, it's going to be so recognizable to many of my viewers. But I also see at a glance that the cholesterol and the LDL are relatively lesser. Yeah. So surprise, surprise.
I think the whole cholesterol, LDL, HDL, it's a big mistake. It's a big mistake we are doing for decades, because it's, the statins, they work, but they do not work because they lower the cholesterol. Because there is a number of statins which have been developed now, which are more efficient in lowering cholesterol, but they have no beneficial effects. So I think we are misinterpreting what is the real value of statins. But this is a different story. I don't want to open the cholesterol war. But my personal opinion is we are just wrong. Because, the cholesterol level is, I think, over 95% genetic. It has nothing to do with what you eat. It's your genes and it's some other things, but not the diet. But okay, leave the cholesterol out. So the other thing is, what about diseases? So we looked in people with many different diseases. And in most diseases, what we see that ill people look older than the healthy people. And it doesn't matter which disease we are looking at. It could be lupus, colitis, diabetes, even colorectal cancer. People who are ill look older. So we do not have a biomarker of a specific disease, but we are seeing a molecular process which is involved in many different diseases. And what we think, it's really a low-grade chronic inflammation, which is underlying many complex diseases we have now. And then the second thing was, we looked into the world. I asked all my friends, can you collect a couple of hundred samples from the region where you live? And then we collected 27 populations from all around the world, from Papua New Guinea, Africa, Asia, Europe. And people in different countries age with a different pace. So what we see that the glycans between countries are different. And the thing which explains the difference the most is actually expected lifespan. and the GDP of a country. So if you live in a poor country with a low expected lifespan, your glycans move to old glycans much faster than if you live in Western Europe or some other countries where the expected lifespan is much higher. So it's a environment where you live affecting your immunoglobulin glycans and making them move to this older shape. And of course, the question is, what is here a cause and what is the consequence? So if you are aging faster, are your glycans just following, or are glycans actually making you age faster? And this is something which is really complicated. It's not easy to answer. We don't have a final answer. We have some hints. So we did a huge pleiotropy study. And pleiotropy is the phenomenon when the same gene affects a number of different traits. So we looked at genes which regulate IgG glycosylation, and through a series of genome-wide association studies, we identified over 40 genes which are in this network. And we looked whether these genes are risk factors for some known diseases. And there are some very obvious ones like Crohn's disease, IBD, asthma, Parkinson. But altogether, we have 94 different phenotypes where things like HDL level, lupus, even cholesterol is regulated by genes which regulate IgG glycosylation. So this does not mean that every of these functions is affected by IgG glycosylation. It just means that there are same genes regulating them. But in some of them, most probably, IgG glycosylation will be before the final disease. So the glycans will change and then you will become ill. Yes?
Hey guys, just a quick reminder that I do need to be supported to provide all of this content, and you can do so at patreon.com/Ivor Cummins, or for a one-off or monthly at PayPal, and you can get that at tinyurl.com/Ivor Cummins. So hopefully you can help me to continue to provide good, correct scientific interpretation and counter somewhat the biased corporate version of science. Thank you, guys. No, I'm just seeing again all the old suspects here, Gordon, but something just triggered in what you were saying. So the Mendelian randomization experiments to prove that genetic cholesterol level that shows that cholesterol is causal rather than associational. Well, there's pleiotropic effects there, just as you described. So someone with a genetically destined higher cholesterol who's destined to have maybe slightly higher heart disease, they could also have this whole mechanism that's really more important than the cholesterol.
Could be.
Yeah,
Could be.
Just curious.
So we tried Mendelian randomization, but our data set is still too small. You need hundreds of thousands of people, and we tried, we didn't see anything, but we still don't have the sufficient power by the numbers. But what we did, we also looked at patients with diseases. So we had patient cohorts with the different diseases, and this is a review article published 2 years ago. And we see that in most of those diseases, glycans are changed. So people, when they're ill, they have this change in glycans. And in some of them, well, actually in all of them where we had appropriate samples, glycans changed before the disease was diagnosed. For example, in rheumatoid arthritis, your IgG glycans will move to this pro-inflammatory direction even 10 years before you get initial diagnosis. In diabetes, it's not IgG glycans, it's a different glycans, but also we see the change in glycans 5 to 10 years before the glucose get affected. So what we think here is really that we have a way to look into the modifications of a protein function through glycosylation, which contributes to the disease development, but we see it only at a very early stage because diseases usually get diagnosed at a very late stage. But I will come back to that also later in the talk.
Oh, just one point on that. Yeah, so I love this. So the reality is that way earlier than diagnosis, the glycans and glycosylation is acting as a biomarker of something that's gone wrong prior to that. And also, it's part of the engine of driving the disease forward mechanistically.
Yeah.
Yes.
Yeah. So one example we have is cardiovascular diseases. For example, we did two large cohorts. One were the twins from Tim Spector, and the other one was the Orkney and Shetland Islands from Gene Wilson, where he collected population samples from thousands of people. And what we see that the glycome composition highly associates with the cardiovascular disease risk score. So the glycans you have now are predictors of heart attack or stroke in the future. But this is only correlation. But then we collaborated with a colleague from UT Southwestern, Phil Scholl, who have shown that IgG glycans, those pro-inflammatory IgG glycans, can actually cause hypertension in obese mice. So if you feed mice a high-fat diet, they develop hypertension. But if you take immunoglobulins from these mice, put them into healthy mice, they also develop hypertension. And then we teamed up and we did together a series of experiments, but he was able to prevent this by feeding mice N-acetylmanoxamine, which is a precursor of salicylic acid. So he was feeding mice with the manoxamine, and then the salicylic acid levels went higher. And these mice were getting obese, but they were not developing hypertension. So it's a very interesting thing. You can just have a single precursor of a glycosylation, feed mice with it, and fat mice do not develop hypertension. So we tried with humans. We did one cohort of humans, but in healthy humans, the feeding with MANAC does not change IgG glycosylation. Maybe if this is decreased in obesity, maybe this would work, but we didn't have a cohort of obese people taking MANAC. But at least in mice, this is working. And what we see in humans, we see associations. So people who have higher blood pressure, who will develop higher blood pressure, they have lower level of IgG cellulation. And this is something we see before hypertension develops. And this was done in a series of studies with Wei Wang, mostly in China, where we saw that people with hypertension have these changes in glycans, but also people with pre-hypertension are already on this trajectory of changing glycans. And
and there's a linear relationship as well. So there's that relationship which infers causality, also a dose response type relationship.
The problem is that this is also correlated with many different confounders. Yeah. So it's very hard to distinguish what is really causal here. But this is just a set of indications which altogether suggest that this might be the case. And one very nice experiment we published earlier this year, there was a big EPIC cohort collected in Europe over 20 years ago. And they collected over 300,000 people. We got access to the German subsample, which had 27,000 people, which were followed for 10 years after the sample was collected. In this follow-up period, there was 508 cases of incident cardiovascular diseases. So people who were healthy when the sample was collected got either heart attack or stroke later. So we looked at glycans, IgG glycans, to see how much they can actually predict future events. And we see that in men, this is glycans, just this single molecule, glycans attached to immunoglobulins, they are as predictive as the entire AHA score, which is the currently used index where they put everything in, age, smoking, everything we can put in. And in women, a single glycan is even more predictive. So these glycans, which we were finding to be causal for hypertension in mice, they seem to predict future cardiovascular events in humans. So of course, this is the first study. This all needs to be replicated. We are currently doing 2 large replication studies with a colleague from the Harvard University. But it does seem that these immunoglobulin glycans, which we know are regulating low-grade chronic inflammation, also participate in the future cardiovascular events, which is kind of big. So based on all of that, we have this GlycanAge test, which quantifies these glycans. So it measures these 24 molecules on IgG, which we know they're effectors of aging. We know that they integrate genetics, epigenetics, and environment. So we have a test which is putting this all together and is measuring molecules which actually do some damage. And I think the key question here is, if we improve our glycans, will we be healthier? So is improving glycans a way to have a healthier life? And we don't have a final answer to that question. We have some indications which I will present you in the next few minutes. So the first question is, can I change glycans by decision? Can I do something to change my glycans? And we don't have many people which have been tracked for a long time. And the person who has been tracked for the longest time is me. So I'm testing myself for over 5 years now. And actually, my glycans are horrible. I think my lifestyle of living too quickly is kind of reflecting in glycans. So I'm over 20 years older than I'm chronologically. And I'm getting older and older. So as time is passing, it's going bad. And there was something here and something here which changed the trend. And of course, when I learned this, I started to try to improve it. And what we know it's sleep, it's stress, it's physical activity, it's diet. These are 4 factors which affect it. So of course, first thing, I was trying to sleep more, easy thing. Didn't really help much. I tried to exercise more. I was really exercising a lot for some time, and it didn't help much. So just doing a lot of physical activity does not improve. I can't really control stress, so I cannot say how much the stress is a factor, but I could control diet. And these 2 things here is when I lost over 10 kilos. So I went on a diet, lost extra weight, and I do have probably 20 kilos too much. And when I improve, it's going down. The problem was I always gain my weight back. I can lose it, but when I stop, well, usually I lose it on holidays, and then I go back and work and stress and everything, and I get it back. So I'm on this bad trajectory, and the only thing I can do, I can be hungry. And this is the famous caloric restriction monkey. This guy is on a diet for 20 years. He's healthier, but not really happy. So I have to decide in being happy or healthy, which is not an easy decision. But this is me. So what we know about other people. So generally, weight loss works. So we did several cohorts of people undergoing bariatric surgery. They lose 20, 30 kilos after that, and their GlycanAge improve. We had a person improving for 30 years within 6 months. But this is drastic. Not everybody will go on a bariatric surgery. So we also looked at, in Twins UK cohort, people have been collected at multiple time points, the samples. So we looked at 2,000 twins collected in 3 time points across 15 years. And what we see here, so these are the pro-inflammatory glycans, these are the anti-inflammatory glycans. So they go up with age in all of them, but if they are losing weight, they go up slower than if they're gaining weight. So twins who were gaining weight were kind of aging on a GlycanAge scale faster than twins who were losing weight. So definitely obesity is something which is driving the pro-inflammatory IgG glycome and is bad for the GlycanAge.
Oh yeah, just on the obesity there, this is an old chestnut. So is it the obesity per se that's driving it, or is it what is driving the obesity is driving it?
We don't know. So one experiment we were planning before the corona set in, so it's now kind of delayed. We wanted to look into this fat removal, where they go for aesthetic surgery, just removing the fat, to see that there's liposuction. whether this would have an effect, because it is known that fat can be pro-inflammatory. So it could actually only be the excess of fat tissue which is causing the damage. So if you just remove it, maybe it will get better. But if it would not, then this would mean that it is maybe what is causing the obesity is also causing the glycans. So we don't know.
Exactly. And I just have a couple of things to mention there. So I know the visceral fat, which is kind of very interacting with the immune system. They have done studies way back with mice, I think, and they've removed visceral fat in high fat diet mice. And the results were kind of ambiguous. There was some indication it was affecting metabolic health, but the real one, which has stuck out for me for many years now, is the metabolically healthy obese study. And they had guys who were all 45 BMI. So these are super big guys. And there was a subset who were insulin sensitive. And they had really good biomarkers across the board. And then there were the insulin resistant super obese. And their blood markers looked shockingly bad. But the really striking thing that came out of it when they investigated is that in terms of their insulin resistance, you could predict their insulin resistance or sensitivity, which is a huge factor in disease, 98% predictable with just two measures: the serum adiponectin and the macrophage content of their visceral fat. So those two measures gave a straight line. So I think there's obese that are really healthy, even though they're super obese. And then there's, of course, generally obese are really unhealthy. But I think it might point to the causality. You know, you can have safe fat, safe obesity, but it depends on the metabolic health underlying, which I'd say is intimately tied, of course, into the glycans.
So the biggest problem we have in understanding anything in science is that people are different. And exactly as you said, somebody can be obese and healthy, somebody can smoke for 80 years and be healthy. So it's we still are lacking the biomarkers which will tell us, you know, am I a person who can be fat? You know, can I enjoy food and still stay healthy? Or am I in the majority who should not do it? So I think we still need a lot of research. I hope that these glycans will help people get more information, and combining glycomics with the genomics or epigenomics, eventually we will know. But for now, we are still learning. And I have a few more slides about what we are learning. We learned that exercise can be beneficial. For example, in one study of high-intensity interval training, we showed that it's immediately beneficial, but many times exercise can actually have pro-inflammatory effects. So usually when people exercise too much, and this was a study of ladies preparing for the bikini fitness competition, they get more pro-inflammatory. So exercise is something which should be kind of optimized. Not just telling people just go and exercise. It should be the proper way of exercise for everybody. So we don't have a magic recipe yet, but just go and run is not a solution. And we have an interesting phenomenon is that we collaborate with Joseph Raphael in New York. He has a kind of anti-aging clinics, and most of his clients are 20 to 40 years younger. So what he does actually improves GlycanAge, but they do very aggressive treatments, all the growth hormone, testosterone, all the supplements. It's not something you would just recommend to everybody, but there are these biohacking approaches which could help, but I think they require a lot of research. There are some people who are younger just by lifestyle. For example, this is my wife. She's over 20 years younger than her real age. So she's half my age in GlycanAge.
Wow.
And she doesn't do any of these aggressive things. She tries to live healthy. Something also we learned is estrogen is a very important factor. There was a study in US where menopause was chemical induced, and these ladies got on medium 30 to 40% older in a couple of months. So they got 10, 15 years older just when the hormones were blocked, and the estrogen replacement prevented this. So those who were receiving estrogen replacement, they stay the same, while the ladies without estrogen, they got much older. So
And
obviously,
oh yeah, again, sorry to interrupt, but and that may speak to as well the whole thing with postmenopausal women. Their cardiovascular risk rapidly over 10 years past menopause gets up to men's levels. But before menopause, they're relatively highly protected against this kind of metabolic chronic disease of heart disease. So yeah.
and not and many other things, and we do think that the glycans are part of this story. Not everything, but they're probably IgG glycans are molecules which contribute to this increased risk around the menopause.
Yeah.
And coming close to the end of my presentation, as just reminding you and everybody else, is that glycans are molecular effectors. They do the work. So they change the structure of many proteins. And if we want to understand them, we have to look the entire molecule, bird with the feathers, protein with the glycans. And this is something what we are trying now to do with this GlycanAge story. The GlycanAge actually is something we already commercialized. So we translated this science into something people can go and buy. It's measuring IgG glycans. Most probably, we will soon also come out with the metabolic GlycanAge, which is measuring other forms of glycans, but this is still only in the research arena because we haven't. So the biggest problem with the GlycanAge was to make the test doable, because we were doing the test on immunoglobulin, which you have to analyze from blood. So you have to collect blood from people. And what we managed to develop a test from dried blood stains. So you can just do a finger prick, collect few drops of blood and mail it. And this is something which works for GlycanAge fine at the moment. We are at the moment in a process of developing this for this metabolic GlycanAge, because this is mostly inflammation, chronic inflammation, aging. This will be more metabolic aging, diabetes development, and so on. But the general idea is that we develop tests which will identify people early on a trajectory of disease development. Because normally, we go to hospital when we are ill. Ill means something is really hurting us. You know, there is a pain and we cannot function. And then we go to hospital, and then we got diagnosed, probably based on location and some symptoms, we got a diagnosis. And then we got the therapy. And most of these diseases are called chronic because you never get cured. You only get, your symptoms are decreased and you continue to be ill for the rest of your life. Hopefully, some early reprogramming therapy could help. This is kind of modern medicine. You identify it early, you give reprogramming medicine, and then people can get healthy. But if we catch them here, before there is a disease, if there are only biomarkers directing the disease development, which we can identify, and maybe, and for some of them we know, we can change them with a simple lifestyle intervention. You know, you lose a couple of kilos, you maybe change your diet, or you exercise a bit more, then we can prevent the disease. We can keep people in the healthy area for longer. So what we are hoping that these biomarkers will give an early warning sign. You're not ill, but you're on a way to become ill. Try to change it, see whether it works, and stay healthy. Because what we are measuring are effectors. These are molecules which affect multiple aspects of the immune system. And this is still hypothesis, but we believe it is true. If you improve your glycans, we will also decrease the risk of a disease. Of course, we don't know it yet because we have to wait for people to develop disease or not. But I think what is important is that we know that there are some healthy habits which can help you stay healthy, but people don't do it. And people generally don't do it because they don't get the feedback. You know, I don't want to do something now to see the effect after 20 years. There's no motivation into that. So what we are hoping here, that this test which we develop can give people a feedback what you are doing is working. And for example, we have a number of customers who improve their GlycanAge, and this made them happy. And they improve it by doing something which we know is healthy. So we are trying to motivate people to live healthier by giving them objective insight into the current state of low-grade chronic inflammation. So this is something which you cannot measure with the regular biomarkers. And then we have a quantitative tool which can evaluate effects of what you are doing. I know if I go on a diet, I lose 10 kilos, my GlycanAge improves. So this is something which people can use to motivate themselves. And at the end, I have to say that what we are doing is costing a lot of money. So we have a lot of research grants funding for it, over 15 million euros of research from different European grants, American grants, Croatian grants. And I have to show that I have a large team of scientists working in my group, and they actually do all the work. I just do the talking. So this would be it for my slides.
Excellent. Thanks so much, Gordon. No, that was superb. And no, I love this thing because it's not just a biomarker, it's also got a causal element. Yet to be fully proven, in fairness. But if so, it'll be very proximate to the disease risk. So a lot of other biomarkers, they indicate something's wrong under the hood, but this one may be much more proximate and directly connected. So we say in engineering, if you don't measure it, it don't get fixed. If you don't measure it, you can't understand it. So measuring is really important. And then when you make your interventions, you've got a really good measurement that tells you their efficacy and motivates you and drives you forward. So this technology is available now, or this is kind of coming. Like the immuno one is actually on the market now.
So technology is available. So this immuno, the GlycanAge test is something what everybody can buy. You can go on a web shop and buy a GlycanAge test. The other technology is also available, but it's available for researchers. So we can collaborate with the people doing a clinical study, because for the other thing, what we need a blood sample. Actually, we need a plasma sample, which you cannot just pick up and mail. It has to be sent frozen. So logistically, it's not convenient to do it for a single patient or for a few people. So generally what we do, we team up with researchers doing cool things. So when I read the paper, which is interesting, I email that person, say, would you like us to look glycans into your cohort? And for example, at the moment in our lab, we have 1,000 people which were on 4 different diets for a year. So we'll know which diet affects glycans in which way. In a couple of months, probably we'll publish that paper. Or we have COVID cohorts coming in now, people with severe and mild COVID. And it does seem that glycans are also predictive there. So it's really cool to have a lab. We generate so much data and then collaborate with people who have done a very cool experiments, and then just trying to match these two things.
Absolutely. And I might make a cheeky prediction here that if you put a cohort of people versus default healthy diet and exercise and put them on a low carb or ketogenic diet with fasting, intermittent fasting, and magnesium, potassium, and a few other things, and sun exposure, did a multi-factor intervention, I'm guessing your needle would move a lot, even without much exercise.
So I'm pretty sure. So I think we need some basic exercise to, but over exercising is not much. Intermittent fasting, I think it's very important. And I have a little bit different causality theory than other people. What I say why intermittent fasting is important is that it regulates our microbiome in a way that we evolved with our microbes. We live together forever, for millions of years. And we have a way of selecting microbes we like. We actually feed them with our glycocalyx. So what we do in our guts, on the surface of the endothelium, we just produce a big glycans, which bacteria can eat. But only those bacteria which have enzymes to degrade it. So there was coevolution of bacteria and us. And then you have a modern situation, which did not occur until maybe 50 years ago, that we are eating all the time. And there is always food in our guts. And then bacteria can eat all that food. And then there also the bacteria which will propagate there would be the most aggressive, those which are dividing fastest, and they may not be very good for us. So what is good for us is to get rid of all that food, have only the empty guts, bacteria which can feed on our glycocalyx, they stay, everything else gets flushed away. And when you think about it, every religion in the world has a fasting as one of the things we have to do. So there is, this eating all the time, it's not healthy. High carb, this happened with the agricultural revolution. And the agricultural revolution was the worst thing for the human health in history. If you look at the skeletons, people became smaller and less healthy when they started to live on few grains. So it's there is deep logic in that, but it's there are too many commercial interests in the current diet
that is for sure. I've been battling them for years. But yeah, even the agrarian or agricultural revolution, absolutely a disaster, maybe 9,000 years ago. And not only is the high carb element and maybe less dependency on nutrient dense, really rich ancestral foods like meats and fish, eggs, whatever, and a switch over to this nutrient poor grains. But also the grains are bringing in many cases, lectins and other kind of toxic elements, which in themselves have been shown to damage the glycocalyx, in many of the layer.
Do you know what lectins do? What are lectins?
Oh, lectins, well, in wheat, for instance, there's wheat germ agar-gluten and there's various other gliadin, but lectins are the defense mechanism, is the general understanding of the plant, and bringing them in large quantities.
They bind glycans. So lectins are receptors for glycans. And all the microbes, all the enveloped viruses and the bacteria have a lot of glycans. So plants catch them with lectins. We also have our lectins, which are defense system. We have our mucins, which are kind of a decoy in the mucus, which we then cross-link viruses and these mucins and get all the viruses out of our respiratory tract.
Now, we're going to go. Do you have a little time, Gordan? Because I'm conscious you're on vacation, but there's almost a part 2, or we could circle back, and that's to the current viral situation, which Creon. We had a fantastic podcast on the 5 or 6 layers of the immune system, including the mucosal and what you're referring to. But could we spend a little time talking on the relevance to the current? Well, I'll have to say, Gordan, this has been fantastic, and I think it's going to bring this knowledge that you've worked so hard on with your huge team over the years to a lot more people. I think that's really important. But I am conscious you have a hard stop, and I don't want to impact. So maybe just some last words, and we'll draw to a close.
Oh, it was great talking to you. Thank you for doing this podcast, and I think it is important to really bring this message to as many people as possible, because this is part of science which is in very early stages. It has to be developed further, and the only way to develop it is to get more people involved.
Absolutely. So to that end, hopefully this will help in that process. So thanks a lot, Gordan, and we'll circle back hopefully and catch up again on this.
Thank you. It will be my pleasure.
Great stuff. Thanks for listening, guys, and hopefully you enjoyed it. And just a reminder that I do need support to continue putting together all of this content, and at patreon.com/Ivor Cummins, or for PayPal at tinyurl.com/Ivor Cummins, where you can do a one-off or a monthly support. So I'd really appreciate that, guys, and keep me getting the science out there and countering perhaps the more biased corporate type science. Thank you.